Dear learners, welcome to our first lecture on pharmaceutical biotechnology that deals with modern pharmaceutical development. Whether you are aiming for top score in your pharmacy competitive exam or you are passionate about learn biotechnology subject, this lecture for you. Pharmaceutical biotechnology is not just a branch of science. It's an important role in modern pharmaceutical development. So, pharmaceutical biotechnology is a branch of science that focuses on the use of biological system, organism and biological processes to develop pharmaceutical product such as drugs and vaccine. Pharmaceutical biotechnology develop pharmaceutical product such as drugs and vaccine. Also, this field play a significant role in development of biopharmaceuticals, including monoclonal antibodies, example that is rituximab anti-cancer drug, gene therapy, also recombinant proteins, valuable proteins development, insulin production for diabetic mellitus treatment, also vaccine development, also hormone development, also genetic gene therapy. This technology directly repair or replace defected genes in human cells, treatment of hemophilia. So this all point we'll discuss about in today's section. In that section we learn about syllabus as well as books as well as expected MCQs, multiple choice questions, pharmacy competitive examinations discussion, also previous year question paper discussion. So let's start today's lecture. Pharmaceutical biotechnology objective. So in that first objective, that is understand the importance of immobilized enzyme in pharmaceutical industry. Second, genetic engineering application in relation to production of pharmaceutical. Also importance of monoclonal antibodies in industry. Also use of microorganism in fermentation technology. So first objective, that is understanding the importance of immobilized enzyme. Immobilized means prevention of mobility, movement of enzyme, particular restriction entry of enzymes particular site. So that can help to reuse of that enzyme. Second, genetic engineering. Genetic engineering is helpful for development of insulin, as well as treatment of defected genes, gene therapy. This technology repair defected genes in human cells. Monoclonal antibodies such as rituximab anti-cancer and microorganism fermentation technology for development of various beverages, as well as antibiotics, as well as vitamins, as well as amino acids. So these are the four objectives. First, understand the importance of immobilized enzyme in pharmaceutical industry, genetic engineering application in relation to production of pharmaceuticals, importance of monoclonal antibodies in industry, and use of microorganism in fermentation technology. So this is the subject, a research based subject. Various scientific applications of biotechnology in genetic engineering, medicine, fermentation technology. So first unit, pharmaceutical biotechnology, in that first unit, that is first unit in that brief introduction to biotechnology with reference to pharmaceutical science. Brief introduction to biotechnology with reference to pharmaceutical science. Second, enzyme biotechnology, method of enzyme immobilization and application. Biosensor, working and applications of biosensor in pharmaceutical industries. Brief introduction to protein engineering. Use of microbes in industry, production of enzymes, general consideration, amylase, catalase, peroxidase, lipase, protease, penicillinase. And last one, that is basic principle of genetic engineering. So in first, brief introduction to biotechnology. Biotechnology that deals with living organism or biological system or biological processes. Pharmaceutical application, in that development of biopharmaceutical substances, insulin, monoclonal antibodies. Enzyme biotechnology, immobilization means prevention of movement of enzyme, reuse of enzyme, that is helpful for. So enzyme, immobilized enzymes used in food industry also, as well as pharmaceutical industry also, for development of drug. So enzyme is act as a catalyst. Next, biosensor. Biosensor is nothing but it is an analytical device used to detect the material. Protein engineering, in that development of valuable proteins. Use of microbes in industry, in that various microorganisms used for development of antibiotics, penicillin, and principle of genetic engineering. This is the last point. This is the first unit. So questions, multiple choice questions asking or expected for competitive examination. That is first, which of the following is the primary application of biotechnology in pharmaceutical sciences? First question, which of the following is the primary application of biotechnology in pharmaceutical sciences? First option A, development of synthetic chemicals. B, production of vaccines and therapeutics. C, manufacturing of traditional medicines. And D, all of the above. Question is that which of the following is the primary applications of biotechnology in pharmaceutical science? So development of synthetic chemicals is not options, correct? Production of vaccines and therapeutics is the correct answer. Manufacturing of traditional medicines means herbal medicines is also not role of applications of biotechnology, pharmaceutical sciences. So production of vaccines and therapeutics, major applications of that pharmaceutical sciences, production of vaccines and therapeutics. Next, which of the following method is not typically used for enzyme immobilization? Enzyme immobilization means prevention of entry or movement of enzymes. So method that is covalent bonding, cross-linking, adsorption on solid support. But question is asked is not method. So genetic mutation of enzyme is the not method. So correct answer is that is D. Next, immobilized enzymes are used in option A, diagnosis or treat the disease. Option B, pharmaceutical. Option C, food preservation. And option D, all of the above. Immobilized enzymes are used in diagnosis, treat the disease. B, pharmaceutical. C, food preservation. And D, all the above. The correct answer that is all the above. Next, which of the following true points about biosensor? So option A, biosensor is an analytical device that detects changes in biological processes and convert them into electrical signal. Option B, it is a device that analyzes the concentration of substance in solution. And C, medical diagnostic, environment monitoring and food safety assessment. So yes, biosensor is analytical device. Second, it is device to analyze the concentration of solution, also monitor food quality, medical diagnosis, environmental monitoring means air quality index. So all of the above is the correct answer. Next question, that is protein engineering involves. Protein engineering, first option A, that is designing and modifying enzymes for industrial applications. B, protein engineering is the process of developing useful or valuable proteins through the design. Option C, both A and B. And option D, none of this. So protein engineering is used for developing valuable proteins, desired proteins, that is the required. So protein is required to growth of body. So well protein is necessary. So that's why protein engineering is the process of developing useful or valuable proteins through the design. Also designing and modifying the enzymes for industrial applications. So both A and B. Next, which of the following microbes is commonly used for industrial production of antibiotics? So microorganism. So development of, option A, that is yeast, B, bacteria, C, fungi, and D, algae. So but which of the following microbes is commonly used for the industrial production of antibiotics? So fungi is the correct answer. Commonly used means development of antibiotics, penicillin notatum and penicillium chrysogesum. Yeast used for fermentation processes. Next, which of the following enzyme is used in detergent industry? Option A, protease, option B, amylase, C, lipase, option D, all of the above. Protease is used, amylase is used, C, lipase also used, D is the correct answer. Next, which of the following points true regarding amylase? Amylase use food fermentation and pharmaceutical industries. Amylase is an enzyme that helps digest carbohydrates. It is found in the saliva of humans. And D, all of the above. A, use in food product development, fermentation and pharmaceutical industry, bakery product food. In that, amylase is an enzyme that helps digest carbohydrate, and C, it is found in saliva of humans. So D, all of the above is the correct answer. Next, which of the following is application of catalase? Food industry, improve the stability of therapeutic proteins and drugs. B, C, both A and B, and D, none of these. So both A and B is the correct. Next, which of the following techniques is central to genetic engineering? Option A, polymerase chain reaction, B, electrophoresis, C, microscopy, and D, NMR spectroscopy. Microscopy, identifications of particle size. NMR spectroscopy, it provides the signal. NMR signals molecule, how much signals, frequency. So this two option is cut. Electrophoresis, measure that is polymerase chain reactions. So option A is the correct. Polymerase chain reactions is the identifications of infectious disease as well as criminal investigations. This is also role of polymerase chain reaction. Okay, these are all questions, that is unit 1. Okay, let's discuss unit 2. Study of cloning vector, restriction endonuclease and DNA ligase, recombinant DNA technology, application of genetic engineering in medicine, applications of rDNA technology and genetic engineering in the production of interferon, vaccine, hepatitis B, hormones, insulin, and last point, that is brief introduction to PCR, polymerase chain reaction. So these are the syllabus of Unit 2, study of cloning vector, restriction endonuclease and DNA ligase, recombinant DNA technology, applications of genetic engineering in medicine, applications of rDNA technology and genetic engineering in the production of interferon, vaccine, hepatitis B, hormones, insulin, brief introduction to polymerase chain reaction. So questions, multiple choice questions asked or expected for competitive examination. Which point true about cloning vector? So cloning vector is a vehicle which can transmit the genetic information from one to another. So first option a small piece of DNA take from virus, plasmid or cells of the higher organism that can be stably maintained in an organism. Ability to carry large DNA molecule. Plasmid, bacteriophage, cosmid are the types, and D all of the above. So cloning vector is a carrier ability to carry large DNA fragment, or is it act as a vehicle to transmit the genetic information from one to another. That is a cloning vector. Cloning means development of multiple copies. So option D is the correct. Next, what is the primary function of restriction endonuclease recombinant DNA technology? First A, ligate DNA fragment. B, to separate DNA molecule by size. C, to cut DNA at specific sequence. And D, to replicate DNA molecules. So restriction endonuclease or restricted enzymes, endonuclease enzymes. Enzyme is role that is cut, catalyst, breakdown. So restricted endonuclease means those enzymes which cut the DNA at specific sequence, specific site, recognized site. That is the C option is the correct. To cut the DNA at a specific sequence. Third question, DNA ligase is used to option A, break down the RNA strand. B, join the two fragments of DNA from a longer strand of DNA by passing them together. C, unwind double stranded DNA. And D, synthesize RNA from DNA template. So DNA ligase is used for join the two fragments of DNA from longer strand of DNA by passing them together. So B is the correct answer. Next, MCQ 4. Which of the following is an application of genetic engineering in medicine? First, genetic engineering. Genetic engineering is a technology used for development of valuable proteins. Protein engineering develop the valuable proteins. Genetic engineering development of insulin. First option is that is creating genetically modified crops. B, producing human insulin from recombinant bacteria. C, developing biodegradable plastic. And option D, production of biofuels from algae. So creating genetically modified crops. Medicine, question is asked, that is applications of genetic engineering in medicine, not asked in agriculture. So creating genetically modified crops, yes, in agriculture role, but here asked in medicine. Developing biodegradable plastic, producing biofuels from algae. So mainly in medicine, production of human insulin from recombinant bacteria. So B is the correct answer. So insulin is used in the treatment of diabetic mellitus. Next, recombinant DNA technology is used to produce interferon for. So interferon is a proteins, is antiviral. Treating bacterial infections. Option B, enhancing the immune response in cancer and viral infection. C, stimulating tissue growth. And D, enhancing the production of red blood cell. So option B is the correct answer. Enhancing the immune response in cancer and viral infection. So interferon is a proteins. It prevent the viral infection, antiviral substance. Next, recombinant DNA technology is used in the production of which of the following vaccine? Chickenpox, option A, option B hepatitis B, option C influenza, option D malaria. So option B is the correct. Hepatitis B vaccine is developed by rDNA technology. Question number 7. Which hormone is commonly producing using recombinant DNA technology in bacteria? Option A thyroxine, option B insulin, option C growth hormones, and option D estrogen. Option B is the correct. Insulin is developed from rDNA technology bacteria. Now, next, polymerase chain reaction is primarily used for option A amplifying DNA segment, B cutting DNA into small fragment, C sequencing DNA, D ligating DNA molecules. So option A is the correct, amplifying DNA segment. Next question, polymerase chain reaction can be used for diagnosing genetic disorder, cloning genes in bacteria, cloning genes in virus, and D all of the above. So polymerase chain reaction can be used for diagnostic genetic disorders, not cloning genes in bacteria, not cloning genes in virus, and D not all of the above. So diagnosing genetic disorder. So next, unit 3rd syllabus, that is types of immunity. Humoral immunity, cellular immunity, structure of immunoglobulins, structure and functions of MSC, hypersensitivity reaction, immunostimulation and immunosuppression, general method of preparation of bacterial vaccines, toxoid viral vaccines, antitoxins, serum immunoblood derivatives and other products related to immunity, storage conditions and stability of official vaccines, hybridoma technology, production, purification and application. So these are the syllabus of unit 3rd, types of immunity, humoral immunity, cellular immunity, structure of immunoglobulins, structure and functions of MSC, hypersensitivity reactions, immunostimulation and immunosuppression, general method of preparation of bacterial vaccine, toxoids, viral vaccines, antitoxins, serum immunoblood derivatives and other product, storage condition and stability of official vaccines, hybridoma technology, production, purification and applications. So expected MCQ, that is multiple choice questions. First, that is which of the following is responsible for the production of antibodies in humoral immunity? Option A is T helper cell, B cell, option B, option C cytotoxic T cell, and option D macrophages. So immunity is used protect from foreign particles. So first question, that is which of the following is responsible for production of antibodies in humoral immunity? So option B is the correct. B cell is responsible for humoral immunity. Second, the basic structure of immunoglobulins molecule consists of option A 2 light chains and 1 heavy chains, option B 2 heavy chains and 2 light chains, option C 2 light chains and 2 heavy chains, each forming Y-shaped structure, 1 light chain and 1 heavy chains, D option. So option C is the correct, 2 light chains and 2 heavy chains, each forming Y-shaped structure. Next, the main functions of major histocompatibility complex MSC molecule. So MSC molecule is a recognized self and non-self molecule. To identify self and non-self molecule, first option A, that is to help antigen presentation to T cells, B to produce antibodies, option C recognition and help to immune system recognize foreign substance, and D to produce memory cell. So option C is the correct, recognition and help the immune system to recognize foreign substance. identifies self and non-self molecule. That is the role of MSC molecule. Next, type 1 hypersensitivity. Type 1 hypersensitivity reaction is mediated by hypersensitivity allergic reaction. IgG, option A, option B IgE, option C GA, and option D IgM. So option E is the correct. IgE, it causes hypersensitivity reaction type 1, immediate hypersensitivity reaction. Next, which of the following is a immunosuppression? Immunosuppression means suppress the immune system. Option A, administration of cytokines. B, administration of corticosteroids. Use of adjuvant in vaccine. D, immunization with live attenuated vaccines. So use of adjuvant in vaccine. Vaccine is a stimulatory immune system, and that is not correct answer. Option C and D is eliminated. And administration of corticosteroid is the correct answer. It can suppress the immune system, reduce the performance of immune system. That is the role of steroids. Next, question number 6. Which of the following is a primary method used for preparation of bacterial vaccines? Using live attenuated bacteria, using synthetic peptides, using bacterial toxins, and D, using bacterial DNA. So following primary method used for preparation of bacterial vaccines, using live attenuated bacteria is the correct answer. A is the correct answer. Next, the ideal storage temperature of most of vaccines. Option A 2 to 8 degrees Celsius, option B 10 to 20 degrees Celsius, option C 20 to 80 degrees Celsius, option D 20 to 25 degrees Celsius. So option A 2 to 8 degrees Celsius. Cold temperature, refrigerator temperature is required to vaccine stability. So this can stable vaccine stability. It affects the safety of drug. So drug is stable, then that is the safe. If you not stable, that is not safe. So stability is helpful for safety of drug. Physical, chemical, therapeutic, and microbiological drug substances are those stable. That is stability. Next, hybridoma technology is used for the production of A, antibodies, B, bacterial vaccines, C, enzyme inhibitors, and D, cytokines. So monoclonal antibodies means antibody development is the role of hybridoma technology. Hybrid, synthetic, the technology which developed by antibodies. So that is option A is the correct. Next, unit 4 syllabus, immunoblotting techniques. ELISA, enzyme linked immunosorbent assay. Western blotting, southern blotting. Microbial genetics including transformation, transduction, conjugation, plasmid, and transposition. Another introduction to microbial biotransformation and applications. And last, mutation, types of mutations and mutant. Genetic change mutation, immunoblotting techniques, microbial genetics including transformation, transduction, conjugation, plasmid, and transposition. Introduction to microbial biotransformation and applications, mutation, types of mutations, and mutant. So these are the syllabus of unit 4. Questions expected, multiple choice questions in the 4th unit. First question, that is the Western blotting techniques is used to detect in a sample. The Western blotting techniques is used to detect. Option A, specific proteins, option B, enzymes, option C, nucleic acid, option D, polysaccharides. So Western blotting techniques is used for specific proteins identification. You have to remember. ELISA techniques is used to detect. Enzyme linked immunosorbent assay techniques is used to detect A, antigen or antibody. Test for HIV, C, both A and B, and D, none of these. So ELISA is the techniques which detect antigen or antibody as well as test for HIV identification. So option C is the correct, both A and B. Next, Southern blotting is primarily used to detect. Southern blotting is primarily used to detect A. Proteins, B. Specific DNA molecules, C. RNA molecules, and D. Antigens. So Southern blotting is used for specific DNA molecules. Option B is the correct, specific DNA molecules identification, Southern blot. Next, question number 4. The process by which bacteria take up foreign DNA from their external surrounding or environment is called A. Transduction, B. Conjugation, C. Transformation, and D. Transposition. Question is that the process by which bacteria take up foreign DNA from their external surrounding is called transduction, conjugation, transformation, and transposition. Transformation is the correct answer. Next, question number 5. In conjugation, genetic material is transferred from one bacterium to another through direct contact. Option A, transduction, B, conjugation, C, transformation, and D, transposition. So option B is the correct, conjugation. Next, transduction involve the transfer of genetic material in bacteria. Question A, option A, plasmid, B, fibri, C, viruses and bacteriophage, D, ribosome. So C is the correct, viruses, bacteriophage. Next, question number 7. Plasmid are option A, small circular DNA molecules and independent of chromosomal DNA. Option B, large linear DNA molecules. Option C, proteins involved in cell metabolism. And D, virus particles that infect bacteria. So plasmid, cosmid, and bacteriophage is the type of cloning vector that transfers the genetic information from one to another. So option A is the correct, small circular DNA molecule independent of chromosomal DNA. Next, transpositions. Transpositions are also known as option A, jumping genes, B, viral DNA, and C, fungal DNA, and D, plasmid. So option A is the correct, jumping genes. Transpositions are also called jumping genes, means extra characteristics of substance. Maize, example, that is maize. Jumping genes, transpositions, that is the jumping genes, the extra characteristics that show the material. Next, microbial biotransformation often used for synthesizing synthetic antibiotics, break down complex chemical into the simple compounds, producing monoclonal antibodies, and D, creating new bacterial strains for vaccines. Microbial biotransformation. Biotransformation means metabolism. Metabolism means conversion of one chemical form into another chemical form with the help of enzyme. Enzyme act as a catalyst. So question that is microbial biotransformation often used for synthesizing synthetic antibiotics, break down complex chemical into simple compounds, producing monoclonal antibodies, creating new bacterial strains for vaccines. So biotransformation is the metabolism. Metabolism means conversion of one chemical form into another chemical forms, means complex to simple. So option B is the correct answer, breaking down complex chemical into simple compounds. Next, applications of microbial biotransformation is the production of synthesis of complex molecules, steroids, production of testosterone and estrogen. Option C, both A and B, and option D, none of these. So option C is the correct, both A and B. for synthesis of complex molecule steroids as well as production of testosterone and estrogen is the role of microbial biotransformation. Next question, that is a mutation, a point mutation that changes codon to stop codon is known as A. Silent mutation, missense mutation, C. nonsense mutations, and D. frame shift mutation. Mutation, genetic change, sudden change of genes, that is the mutations. So mutation that changes codon to stop codon is known as A. Silent mutation, B. missense mutations, C. nonsense mutations, and D. frame shift mutation. So option C, nonsense mutation, is the correct answer. Next, a mutation in which one base pair is replaced by another base pair is called. A mutation in which one base pair is replaced by another base pair is called A. frame shift mutation, B. substitution or point mutation, C. insertion mutation, and D. deletion mutation. So option B is the correct, substitution or point mutation. The mutation in which one base pair is replaced, means changes, by another base pair is called substitution, means changes, point mutation. Next, 13 questions. The insertion or deletion of nucleotides in a gene sequence is typically associated with. The insertion or deletion of nucleotides in gene sequence is typically associated with option A, point mutation, B. frame shift mutation, C. silent mutation, and D. missense mutations. So frame shift mutation is the correct answer, B. 14. A mutation that does not result in any change in the protein sequence is called. Means there is no change, no result in any changes in the protein sequence. Option A, frame shift mutation, B. missense mutations, C. silent mutations, and D. nonsense mutation. So mutation that does not result in any changes in protein sequences is called silent mutation. C is the correct answer. Next, 15. A mutation that causes changes in one amino acid of the proteins is known as. A mutation that causes a change in one amino acids of the proteins is known as. A mutation that causes change in one amino acid of proteins is known as frame shift mutation. So this is the all of the questions, that is unit 4. Next, turn to the unit 5. In that, fermentation is a metabolic process. Fermentation's methods and general requirements, study of media, equipment, sterilizing methods, aeration process, stirring, large scale production of fermenter design and various control, study of production of penicillin, vitamin B12, glutamic acid, blood product collection, processing and storage of whole human blood, dried human plasma substituents. So this is the unit 5th syllabus, fermentation method and general requirements, study of media, equipment, sterilization methods, aeration process, stirring, large scale production of fermenter design and various control, study of production of penicillin, vitamin B12, glutamic acid, blood product collection, processing and storage of whole human blood, dried human plasma and plasma substituent. So let's discuss with multiple choice questions. First, that is, which of the following is not a requirement for successful fermentation process? Which of the following is not a requirement for successful fermentation process? First, A, proper temperature, adequate aeration, means air, C, sterile environment, and D, high salinity. So option D is the correct answer, because proper temperature is required, adequate air is required, sterile environment also needed for free from microorganism. Next, question number 2. In fermentation, the process of sterilizing the medium is important. Option E, prevent the contaminated by unwanted microorganism, B, enhancing growth of desired microorganism, C, increase in nutrient availability, and D, all of the above. So fermentation, the process of sterilizing means prevent the contamination or free from microorganism. Option E is the correct answer, prevent the contamination by unwanted microorganism. Next, third, which of the following sterilization method uses steam at high pressure to eliminate microbial life? Which of the following sterilization method uses steam at high pressure to eliminate microbial life? A. Filtration, dry heat sterilization, C. Autoclaving, and D. Radiation. So filtration is the process for filtration is a unit operation which is used for purification of water, purification of substance, as well as purification of water. That is the filtration process. Dry heat sterilization, autoclaving, radiations. Option C is the correct. Autoclaving. Autoclaving also called as steam sterilization. Sterilization free from microorganism. Question number 4. The type of fermentation vessel that is commonly used for industrial scale fermentation is called? Batch fermenter, continuous fermenter, fed batch fermenter, D. Stirred tank fermenter. So type of fermentation vessel that commonly used for industrial scale fermentation is called stirred tank fermenter. D is the correct answer. Question number 5. Large scale fermentation, the main purpose of cooling system is to. Large scale fermentation, the main purpose of cooling system is to A. Maintain the required temperature for microbial growth. B. Add nutrient to the medium. C. Increase aeration. Control the pH of culture medium. So large scale fermenter, the main purpose of cooling system maintain the required temperature for microbial growth. Next, the process of controlling pH during the fermentation is necessary to. Option A. Prevent the growth of unwanted microorganism. B. Optimize the metabolic activity of microorganism. C. Reduce the oxygen demand, and D. All of the above. B. Optimize the metabolic activity of microorganism. Next, what is typically used to monitor and control the oxygen supply in large scale fermenter? Option A. pH sensor. B. Temperature probe. D. Dissolved oxygen DO probes, and D. Pressure gauge. So C is the correct. Dissolved oxygen DO probes, typically used to monitor and control the oxygen supply in large scale fermenter. Question number 8. The industrial production of penicillin is typically carried out in A. Batch fermenter, continuous fermenter, fluidized bed reactor, submerged fermentation system. Batch fermenter. Industrial production of penicillin. Penicillin is the antibiotic used for inhibit the growth of microorganism. So batch fermenter, industrial production penicillin, typically carried out. Next, which of the following is a key function of aeration in fermentation process? Aeration means supply of air, oxygen to aerobic microorganism. B. Control the temperature of the fermenter. C. Remove the toxic gases produced by microorganism, and D. To maintain the pH balance in the medium. So A is the correct answer, to supply oxygen to aerobic microorganism. Next MCQ. Which of the following true points vitamin B12 is produced by? Or source. Following point, true point vitamin B12. So source, that is fish, meat, poultry, eggs, and dairy products, also vitamin B12. Cyanocobalamin B, commonly known, and C. It is synthesized by some bacteria, and D. All. So which of the following true points vitamin B12 in the point we think? That is a source fish, meat, poultry, eggs, and dairy product. Cyanocobalamin, commonly known as. True points vitamin B12. Not think about producing. So fish meat is also source of vitamin B12, is it correct? Cyanocobalamin is commonly known. Yes, this is also correct. It is synthesized by some bacteria. So D is the correct. So in production is prepared from bacteria. Next, which of the following microorganism is commonly used in the industrial production of glutamic acid? Saccharomyces cerevisiae A, Asparagus niger C, Corynebacterium glutamicum and D, Penicillium chrysogenum. So option C is the correct. Next, whole human blood is typically collected using which of the following method? Apheresis, vein puncture, phlebotomy and D, none of the any or no any. So whole human blood is collected vein puncture. Blood collection method, that is vein puncture. Next, dried human plasma is mainly used for blood transfusion, B, protein production, C, clotting factor replacement therapy and D, tissue culture. Dried human plasma is mainly used for blood transfusion, protein production, clotting factor replacement therapy, tissue culture. Clotting factor replacement therapy. Next, plasma substituents are designed to provide long-term source of plasma proteins, maintain blood volume in case of trauma or surgery, stimulate the immune response, D, promote the red blood generation. Plasma substituent. Plasma substituent, example dextran, substitute of plasma for longer duration or maintain blood volume in case of trauma or surgery. So B is the correct answer. Also, longer durations plasma proteins. Next, the typically storage condition for whole blood is 15, 2 to 6 degrees Celsius. A option room temperature, B 20 degrees Celsius and C 2 to 6 degrees Celsius and D 37. So 2 to 6 degrees Celsius is the correct answer. Next, previous year question paper. Pharmaceutical biotechnology, in that questions will ask 5 into 3 marks. Explain basic principle of genetic engineering. Explain basic principle of genetic engineering. Next, B, what is biotechnology? Enlist applications of biotechnology with reference to pharmaceutical sciences. Next question, discuss production of enzymes. Next, explain working biosensor. Next question, give the example of microorganism used in production of amylase, penicillase and lipase. Next, discuss aeration process used in fermentation. Next, describe the principle of Southern blotting. Next, what is cloning vector? Discuss the ideal properties, cloning vectors and write a note on type of cloning vectors in detail. Next, what are hypersensitivity reaction? Classify hypersensitivity reactions and explain in detail type 1, type 2, type 3, type 4. What is hybridoma technology? Discuss production monoclonal antibodies by hybridoma technology and their applications. What is fermentation? Highlight general requirement of fermentation and discuss productions of penicillin by fermentation technology. So this is the 10 mark questions. Next, 5 mark questions. Explain the method of enzyme immobilization. Write a note on polymerase chain reactions. Discuss productions of recombinant insulin. Write a note on ELISA. Explain humoral and cellular immunity. Next, illustrate microbial genetic information, genetic transformations and conjugation. Discuss design large scale production of fermenter. Describe collection, process and storage of whole human blood. What is mutation? Summarize the type of mutation. Write a note on microbial biotransformation. These are the 5 marks questions. Next, second paper. First, that is what is biotechnology? Enlist applications of biotechnology with reference to pharmaceutical science. Next, enlist applications of immobilized enzymes. Next, highlight the use of microbes in industry. Explain basic principle of genetic engineering. Give brief overview of protein engineering. Discuss aeration process used in fermentation. Describe the principle of Southern blotting. 10 mark question. What is RDNA technology? Summarize applications of RDNA technology and discuss production of recombinant insulin. Next, what is hybridoma technology? Discuss production monoclonal antibodies, hybridoma technology and their applications. What are the hypersubstitute reaction? Classify hypersubstitute reactions and explain in detail. Next, what is fermentation? Highlight the requirement of fermentation. General requirement of fermentation and discuss production of penicillin by fermentation technology. Next, 5 marks question. Discuss working applications of biosensor in pharmaceutical industry. Explain restriction endonuclease with example. Write a note on ELISA. What is cloning factor? Cloning vectors. Explain plasmid as a cloning vector. Discuss general method of preparation of bacterial vaccines. Write a note on polymerase chain reaction. Explain the structure of immunoglobulin. Write a note on microbial biotransformation. Describe collection, process, and storage of whole human blood. And what is mutation? Summarize type of mutation. So this type of questions will ask in previous. This is the question paper. Next, last recommended books, latest edition. These are the list of books which recommended by Pharmacy Council of India as well as Savitribai Phule Pune University. B Pharmacy, Pharmaceutical Biotechnology courses. First, that is B.R. Glick and J.J. Pasternak, Molecular Biotechnology, Principle and Applications of RDNA. ASM Press, Washington DC. R.A. Goldset, Immunology. J.W. Godding, Monoclonal Antibodies. J.M. Walker and E.B. Gingold, Molecular Biology and Biotechnology by Royal Society of Chemistry. Jabrowski, Immunology Enzymes, CRC Press, Dengerland, Ohio. S.B. Primrose, Molecular Biotechnology, 2nd edition, Blackwell Scientific Publication. Stanford F.V. Wittenker A. and Hall J.S. Principle of Fermentation Technology, 2nd edition, Aditya Books Limited, New Delhi. So these are the reference books. So what you learn in today's lecture. So our objective that is to learn the syllabus as well as books as well as expected multiple choice questions for pharmacy competitive examinations discussion as well as previous year question paper discussion. So I hope you understand and this video helpful for future competitive examinations. All the best future examinations to you. Unit 1, these are the syllabus. Unit 2, Unit 3, Unit 4, Unit 5. Thank you for listening and watching this video in advance. Keep learning, keep growing. Thank you.